In this experiment, I'm simply counting the frequency of each of the 64 codons within human DNA. My aim is to demonstrate that there is a mathematical balance between the frequency of any codon and the frequency of occurrence of its mirror image codon. In other words, its reverse complementary codon. I'm only looking at a single strand of DNA. I'm not counting from both strands, which would obviously be perfectly balanced. I'm looking at a single strand, and I'm investigating whether within any single strand of DNA there is this strange mathematical balance between the frequency of any codon and its reverse complement. So let's start the counting. We're looking at chromosome 10 of human DNA, and you can see all the codons and their frequencies, and here we have the number of lines being processed. Each line contains 70 letters or 70 bases. As you can see, so already the frequency of any of these codons is closely or approximately equal to the frequency of its complement. In fact, the ratio in all cases is very close to 1. It's also apparent that this balance is present within each section of DNA, rather than it being the first half of the chromosome mirroring the latter half. It's something which is, the mirroring occurs throughout the chromosome. From the beginning, the mirror is present. What form this mirroring takes, I am not sure. I don't know. I don't know whether it's an identical reflection of each section of the DNA, which includes a reflection of the sequence, or whether it's simply a reflection of the frequency alone. One would tend to assume that if the frequency of every codon is matched by the frequency of its reverse complement, then we would expect there to be some kind of area within the chromosome which is perfectly mirroring the other half, as it were. So, for example, the first half of chromosome would mirror the latter half as a reverse complement. However, this is not the case, simply because we can observe this equality or balancing right the way through the chromosome from when we first started the software. So therefore, the mirror seems to be dispersed throughout the entire chromosome. It would be interesting to see if this phenomena also occurs within coding regions of the DNA. There you go, it's completed the analysis of chromosome 10, and as you can see, the frequency of each codon is quite different. However, you can also see how the frequency of the complementary codons match the frequency of their corresponding codon. It's quite an intriguing phenomenon. The ratios all center on 1, approximate to 1. So I'm inviting any explanation for this. It's almost as if the entire chromosome is one massive inverted complementary palindrome. However, we know this is not the case, but it's still intriguing to find out what's going on and why this balance exists.